Richard Roberts, welcome. You were the co-recipient with Phil Sharp of the 1993 prize in Physiology or Medicine for your coincidental discovery of split genes.
Mm-hmm.
I'd like to start by just exploring a few themes with you. If I say the word mentorship, what does that conjure up?
Well, I think to me, it always signifies a Japanese postdoc that I had in my first year as a PhD chemist, who was really quite an extraordinary fellow. He had this wonderful ability not only to sort of tell you what you should be doing, but at the end of it you understood why you'd done it. And I learned more chemistry from him, I think, than from anybody I've ever met previously. I'm certainly certain more than from any teachers that I ever had at school, or even from my professor or anyone like that in formal courses. He was amazing.
Can you pin down what it was that he...
Not really, no. I mean, you know, the end product was that you understood why you were doing what you were doing. And I think that's terribly important in science, so that, you know, then if you understood why you did it, you can do it again. And if all you've done is do it, then you may or may not be able to actually do it again.
And when you come to look for students, what do you look for in them?
Usually enthusiasm, I would say, is the single most important attribute for me. I mean, you know, most of the people who come to me are smart, so I don't really ever think that that's going to be an issue. But I really look for people who have this spark of enthusiasm, the passion. You know, because people who are passionate usually work twice as hard as those who are not.
So they need to be hardworking.
Well, I think in science you have to be hardworking. Yeah, you know, if you see the competition, you realize everybody else in science is absolutely working flat out most of the time, and everybody else is pretty smart. And so, if you want to compete with them, you also have to be pretty smart and to work hard too.
And the last of these themes is how would you describe your ideal lab environment?
That's I think one of the things that I look for in a lab environment is an attitude of cooperation, collaboration. I like to be working with people who it's fun to collaborate with, not who you think are trying to stab you in the back all the time, and not people who are in competitive mode, at least not with you. I mean, I think competition is a good thing, but what is not good is competition within the same group, even within the same lab setting. I mean, I think it's fine to cooperate with compete with people in other institutions. I like the idea of competition where two people may be going at the same problem but going from different angles. I see no benefit in two people going at something from the same angle. But collaboration is absolutely the key.
And are there physical principles you apply to get people in your lab to collaborate?
Probably not. None that I can articulate, let's put it that. Maybe there are, but none that I can articulate. I mean, I always like to make sure that people have their own individual projects that are non-competitive with one another, so that if people really are trying to get an answer from their particular project, that it's perfectly okay to talk to everybody else because they're not competing with the other people.
But do you like a bustling lab or a quiet lab?
Both have their place, so sometimes it's very nice that it's bustling. It's good to have people in close proximity to one another because then they have to talk. And very often talking is the key to success, because sometimes the only time that you really understand why you're doing something, or particularly why you made a mistake, is because you're trying to explain to someone else what you've been doing, and then all of a sudden you realize you don't understand it yourself so terribly well. I mean, I think this is the advantage of doing research in a university environment where you're constantly teaching, and so you're constantly sort of testing your own ideas in a way when you're trying to explain what you're doing to other people. You know, and sometimes you get this aha moment and you realize that you don't understand why you were doing what you've just been doing. Students A lot of students are interested in what you've been doing, and particularly, how you made the discovery, what it was that turned the light on, and for that sort of thing. But some of them actually are interested in, just very more general things. How does it affect your family life? This kind of thing.
Yes, the work-life balance is always a question. Mm-hmm. Looking at your own scientific beginnings, you had what, from your autobiography at least, looks like a fairly unremarkable scientific start. Was there a point at which you suddenly sort of geared up and had a moment of realization?
Well, you say unremarkable. I think if you compare my start with what is possible today, it would start to look quite remarkable, because I got interested in chemistry through making fireworks. I just thought fireworks were fantastic. I thoroughly understand why terrorists like a big bomb. There's something about explosions and bright lights and so on. And when I was growing up, it was easy to do that. You could buy chemistry sets, you could buy the chemicals you needed to make fireworks very easily. There were books that told you how to make them. And a lot of my friends, and I think a number of my fellow laureates, also got their interest in science in a very hands-on manner by making fireworks, by making explosives, by sort of doing home chemistry kinds of things. And that is something that's very difficult to do these days. We've reached this point in society where we feel we've got to protect the kids from anything. We can't let them do anything dangerous. God forbid they should be doing something where they might lose a finger or an eye or something like that.
They're not even allowed to take a bus to school. Yeah.
And so I think we've actually done a great disservice to the young scientists, or the potential young scientists, by not giving them those same opportunities to experiment and do stuff at home. There's nothing more boring in a chemistry class than watching a teacher do something. Much more fun to do it yourself, and especially if you have some leeway to do things that maybe was not in the curriculum and that you just want to do yourself. So, and that was the thing that led me to science, I think, without a doubt, is this hands-on experimental work that I was able to do at home that you simply can't do so easily today. Much, much more difficult to do anything like that today.
So when did the parallel tracks of your home interest in science and your sort of standard educational interest meet?
Probably when I went to university, I would say. At school, I was always, certainly in chemistry, I was always way ahead of anything that was being taught in the classroom, because I'd been buying textbooks at home and doing stuff at home. I didn't find chemistry terribly interesting at school. Certainly didn't care for physics at school. I liked mathematics. I always liked math. And in fact, math was always my best subject. I was much better at that, even than chemistry or anything of that sort. But I had a problem when I was doing mathematics, when it came to distinguishing between pure and applied mathematics. And it wasn't until I got to university that I realized applied mathematics was really pure math, and it was just the way you phrased the question and the way you interpreted the question that made it either applied or pure. And so then I was okay. Once I figured that out, I was okay. All of my life, I'd always thought I was going to be a chemist. And my aspirations at school were really to be an industrial chemist. It seemed to me that this was a place where you were going to constantly be able to actually get to the lab bench and do lab work. But then when I started doing my PhD, I was fortunate, thanks to this Japanese postdoc who really taught me how to do chemistry. I got everything I needed for my thesis in the first year. So after 1 year as a PhD student, I could have easily written my thesis and got the thing. But in England, of course, you have to do it for 3 years. And so that gave me 2 years to really go looking around and browsing around and thinking about other things. And it was during that time that I discovered molecular biology through reading and decided I wanted to be a molecular biologist.
Because you were working on flavonoids.
Yes, right. I was, my PhD thesis, I was basically given a piece of tree from a Brazilian tree called Macarium, and asked to find out what was in it. And we sort of knew that there would be interest in flavonoids, actually neoflavonoids in it. And there were loads. And I was lucky, I've always been very lucky. One of the new compounds was a key intermediate in the biosynthesis of these neoflavonoids that my professor had predicted should exist, but it was unstable when it had come out of other things. But in my hands it was stable, and so it was sort of the missing intermediate, and I had it, and he was very happy, so that was nice.
So then you had freedom to explore. And your move to molecular biology, did you think that you were moving away from chemistry, or were you being a chemist who was becoming a molecular biologist?
Well, I thought I was moving away from chemistry a little bit, but taking advantage of the chemical training. So at the time, if you wanted to sort of proceed to the frontier of chemistry, you had to do one of two things. You either had to go theoretical, because there was a desperate need for a new theory of organic chemistry. You sort of had the Woodward-Hoffmann rules, but you didn't have a lot else. And I didn't have any good ideas as to what one might do theory-wise. And the other was to go biological. I mean, it was clear that there was a frontier of chemistry that was out there in the biological world. And I just found that far more interesting and something that I might actually be able to do.
So that took you to Harvard, and your work there led really to the job offer from Jim Watson, which seems to have been, from my reading, the seminal event.
Yes, maybe it's
maybe not.
It's hard to know. So, when I was a postdoc at Harvard, I discovered nucleic acids and sequencing, and I went to Fred Sanger's lab to learn how to do RNA sequencing. And that really gave me an interest in kind of the structure of biological molecules. Not structure in the sense of 3D structure, but structure in the sense of knowing exactly what all the atoms were within the molecule. And of course, for DNA or RNA, it's just really the sequence, what is the sequence of bases? And that sort of seems something relatively straightforward that I might be able to understand and do. And so I kind of got interested in sequencing and methodology for doing the same. And I actually tried to get back to England. I mean, I'd originally gone to the US thinking I was just going to spend 2 years in the US. As soon as I got there, I realized, having changed fields, that 2 years wasn't going to be long enough to do anything in a new field. And so it lasted, I did 2 years, and then I got appointed as some sort of, I forget what the exact thing was, but it was sort of slightly better than a postdoc. And then I started to look for jobs. And I was told there was a nice job in Edinburgh, which I applied for. But I never heard anything back from them. And then I was offered a job by Jim Watson. Came completely out of the blue. I'd spent a lot of time with Mark Potashny, who was, at the time, I guess, either assistant or associate professor at Harvard. But I taught him how to do RNA sequencing, and we'd become reasonably friendly. And he apparently had spoken to Jim and sort of said, there's this guy Roberts that you should offer a job to. And so he came up to me in a seminar and said, Jim Watson wants to talk to you. And since I'd never met Jim at the time, I sort of said, oh yes, that's nice, and waited, and nothing happened. A couple of weeks later, Mark came along and said, well, Jim says he doesn't know you, so perhaps you could go and introduce yourself. And so I trotted off to his office and said, I'm Rich Roberts, and he basically opened the conversation saying, we want someone to sequence DNA at Cold Spring Harbor, I'd like it to be you. And this was kind of it. I was in his office no more than 5 minutes, and as I was leaving, he said, oh, and by the way, as a formality, you'd probably better come down and give a talk, but don't worry, you've got the job.
Nice, but slightly disconcerting.
Yes. So I went. I was in Jack Strominger's lab, and I went and talked to Jack, and I said, well, Jim just told me he's offered me a job. I assume he's talked to you about it. And Jack says, no, it's the first I've heard about it, right? It's sort of fairly typical of the way that Jim, I think, still does stuff. He has people who he trusts and talks to, and if they tell him they think something is worth doing, then he'll often just follow their advice and do it. So, and then at the same time, the biology department at Harvard asked me if I would be interested in a job, and they offered me something. So here I was, I had 2 jobs in hand, as it were, and the one I really wanted was back in England, but I'd not heard whether I was even being considered. I mean, I never even got an acknowledgement for my letter of application. So it seemed the thing to do. I had a wife and 2 kids. I'd better take one of these jobs that was offered me.
What do you think was creating these job offers? Was it primarily the fact that you had RNA sequencing?
Yes, completely, right, yeah.
It was having the right technology at the right time that everybody wanted.
Yes. Well, I was the first person in the Boston area to sort of be doing the Sanger method of sequencing RNA. And so anybody there who wanted to learn how to do it kind of came along and talked to me, and I showed them how to do it. So yes, it was just I could do something that a lot of other people couldn't do, but that lots of people wanted to do.
So when you found yourself at Cold Spring Harbor, you were then working primarily on sequencing
So when I first went there, what Jim wanted was to have SV40 sequenced. But as soon as I got there, I realized there were 2 other groups already doing that. Walter Feers in Belgium was doing it, and Sherm Weissman at Yale was doing it. And it seemed stupid for a 3rd group to do the same thing. And so this certainly caused Jim some consternation, because this is what he wanted sequenced. And Jim always likes competition.
So having a group internally and doing it as well is great, yeah.
But I wouldn't do that. It just seemed to me a waste of time. I mean, if these 2 other groups were going to get the sequence, it was not as though there was any shortage of things to sequence. I mean, the entire world was out there made of DNA. There's lots of stuff to sequence. But I'd actually, during my last year at Harvard, had heard Dan Nathans give a talk about restriction enzymes, and particularly about how you could use HinD2. Well, actually, they called it endonuclease R at the time. It was a mixture of 2 enzymes that Ham Smith had discovered. And Dan Nathans was using it to map DNA.
So this gave you the possibility of sequencing DNA?
Well, I saw, the reason that RNA sequencing had been developed first was because there were small molecules to practice on. And in DNA, there were no small molecules, but these restriction enzymes gave you the opportunity to make some small molecules of DNA. And so I thought that would be very useful, that one could get the small molecules to practice on. And so I was actually initially interested in developing methods for sequencing DNA. But then the restriction enzymes themselves, it turned out that there was not only the one that Ham Smith had found, but there were others around too. And that I kind of got into the idea that one might be able to just use the restriction enzymes as sequencing reagents, because they sort of gave you, wherever they cut, they gave you 5 or 6 bases at a time. And so this might be developed into a sequencing methodology. And so we started to make restriction enzymes and look for new restriction enzymes. And it became slowly clear they weren't going to be very much use directly for sequencing DNA, useful as intermediate reagents to make fragments. But they were interesting for mapping, and then recombinant DNA came along, and so they were useful in that context. So they just became generally useful reagents for doing much more than sequencing. And
you became a big producer of restriction enzymes, indeed, which of course led to future things, which we'll talk about. Yeah. Your discovery of the split gene nature of adenovirus was something that came quite quickly. And I'd like to just talk about the sort of conceptual side of it. It was presumably, when you joined Watson's lab, fairly inconceivable that genes were not a continuous stretch. So was the indication there that they were discontinuous prior to your first findings, or was it a complete surprise when you started seeing discontinuous sequences?
Well, I mean, it was a complete surprise and completely against the dogma, because, Francis Crick had sort of come out with this central dogma: DNA makes RNA makes protein, and that was based upon everything that we knew about in bacteria and bacteriophages, where you had a contiguous gene, you had a continuous piece of RNA made from it, and you just read the bases off 3 at a time as codons. And it never really occurred to anybody that the arrangement of DNA sequences in eukaryotes would be any different from the arrangement of DNA sequences in prokaryotes. And we got involved in this because I was interested in trying to characterize a promoter, the region upstream of the gene that kind of says where the gene should begin. And we wanted to know if eukaryotic promoters were different from prokaryotic promoters. There was no inherent reason to think that they would be. But nevertheless, until you look and see, you'll never know.
That's interesting, that you chose to look at something that you thought was going to just yield a null result.
Right. Well, I wouldn't say a null result. I mean, we thought there might be at least one or two interesting differences, but nothing really spectacular. We certainly didn't think there would be a spectacular difference when we got started.
So you were investigating it because you could, or because there must have been something about the search that there must be something?
Yeah, I would say we did it because we could. We thought in order to do it, you needed to be able to do a little more DNA sequencing than was possible. And so in one sense, it was pushing technology a little bit. And it was also clearly a doable problem. It was something that could be done, and it would give you an answer. Either these things were the same or they were different. So there was kind of a nice yes-no sort of answer that could come from it. And so, but the methodology for doing it was not straightforward. So how you would identify the 5' end of a message, you knew they had triphosphates at the end. So that sort of gave you a handle from an RNA sequence standpoint. Here was a special feature, a label, if you like, that you could look for so that you could in fact find the 5' end of the RNA. And the idea was to then figure out where that was being located on the DNA, and then just look upstream, and that would be the promoter. And adenovirus seemed very nice for that, because adenovirus was a linear virus, and we knew that transcription early during infection was coming in from the two ends. And so that meant that between the end and the start point of the message, that's where the promoter must be. So you could just sequence basically the ends of that DNA, which at the time, that was a lot of sequence. People were typically doing just a few nucleotides, nothing quite as long as that. So anyway, so we set off to do that, discovered that the amounts of RNA that were present early during infection were simply not enough to get a sufficient amount of the terminal oligonucleotides to actually map the message. And so we turned our attention to late adenovirus messages. And there we knew that they were making huge amounts of message. Essentially once adenovirus infects and you get a good infection going, then they produce almost all adenovirus message, very, very little cellular message being made. So that was nice. And then while we were doing that, caps were discovered, these funny modifications that take place at the 5' ends of RNAs. And so that actually gave you an even better tag with which to look at these messages. And when we started to do that, we also realized that there was a possibility of doing a more interesting experiment than just finding the messages. And that was, we knew that late during adenovirus infection, the genes, there were a whole bunch of genes laid out along the genome. And so there were at least 8 or 10 different messages that one expected. And so the thought was, if you could characterize the short oligonucleotides at the very beginning of these messages, each one would be different. And so what you could do is there were display methodologies that would allow you to actually look at all of these different oligonucleotides at the same time.
So you could map.
And so you could see what was happening during the course of infection, which messages were going up, which were going down. Actually do the kind of functional genomics people now do on microarrays, but this in a much simpler system, and long before one had thought about all these other
long
kinds
before
of
the
things.
term functional genomics.
Yes, yeah. And so, we thought this would be a really neat methodology, and there might be a nice paper out of that. And so we started to do those experiments. And I had a postdoc, Richard Gelinas, who I'd first met when I'd been a postdoc at Harvard. He was a student up there, and he'd come down to join me. Very talented fellow. And so this became his postdoc project to do this sort of functional mapping of the adeno messages. And so when he did the first experiment, and we dreamt up this nice way to actually select for just these termini, the 5' termini. And so we set that up and got it going, and he did the experiment, and instead of seeing 8 or 10 messenger RNAs, he only saw 1. And so, I said, well, you better go back, do it again, and we sort of looked through his notes, and he went back and did it again and got the same result. And so basically, I told him he must have screwed up the experiment, and I would do it and show him how it should be done properly. And so I did the experiment, got the same results, and of course then I believed it at that point. So I guess that was the point at which we knew that there was something interesting and unusual going on. Didn't know what it was, had no idea what it was. But I would say that was the moment when I knew that there was an interesting finding to be made, whatever it might be.
Still no guess as to what it would be, no?
Oh, well, one always has a guess as to what it should be. And the guess was based upon the fact that by in vitro biochemistry at that time, RNA polymerase, the eukaryotic RNA polymerase that Bob Rader had been working on, was not a terribly good polymerase unless you gave it a primer. If you gave it a short RNA primer, then it worked pretty well as a polymerase. But if you were asking it to make things de novo, it really couldn't do it terribly well. It was rather poor. And so we had the idea that perhaps what we were looking at was the end of the primer, and that the primer, the same primer, was being used for all adenovirus messenger RNAs, and perhaps it was just folding differently at different points on the genome. And so this sort of became my favorite hypothesis as to what was going on, and was ultimately the hypothesis that we ended up testing when we did the key experiment. But in the meantime, we were sort of just trying to get good biochemical evidence to prove that what we were looking at was real and not some artifact. As everybody wanted to tell us, it was all an artifact, of course, because it just didn't make any sense. Everybody knew how you made RNA. You've got an RNA polymerase, it just gets in there and does stuff. So we spent almost a year, I would say, gathering more and more evidence in favor of the idea that all of these messages had the same 5' end. But we really couldn't prove it to anyone's satisfaction. Biochemistry is not always a good way to sort of convince people of what's going on. People like a visualization of some description. So
it's a bit abstract, yes.
Yes, well, it sort of requires an act of faith almost, that the biochemistry is being done properly. And it's not always easy to convince people. And, they, especially when something goes against dogma, if you're doing your biochemistry and everybody accepts the basic premises of how things work and the biochemistry says, sure, that's clearly how it works, then it's much easier to accept it. But this, we were saying something that completely went against the dogma.
So something had to be wrong.
So something had to be wrong, yes, and it was us. So anyway, so we spent about a year trying to really come up with a good experiment, and then one morning early in March of 77, we'd gotten into the habit of every Saturday morning we would sit and kind of have a post-mortem on what had gone wrong the week before and why we hadn't had the success that we were hoping for, and then dream up the next set of experiments. And I remember this one Saturday morning, Richard was up at the blackboard and was writing out some horrendously complicated set of experiments we were going to do, and I really wasn't paying as much attention as I should, and all of a sudden it struck me what we should do. And so I said sit down, and I rubbed off all his scribblings and drew out this experiment that I thought would be a nice experiment to do. And the only problem with it was an electron microscopy experiment, and neither of us were electron microscopists. But fortunately, we had a couple of colleagues down the hall, Tom Broker and Louise Chow, who were just superb electron microscopists. I mean, just as good as they come. And so after I'd drawn out the experiment, Richard kind of said, yeah, that would be a great experiment if it works. And so we went down and talked to them and said, if we make the reagents, could you do the experiment? And they said, sure. Didn't know it would work, but it sort of looked as though it should. No one had quite done it that way before. And so we made the reagents, and on the Tuesday morning they did the experiment, and the very first molecule they looked at in the EM looked exactly the way I'd drawn it up on the blackboard, with one difference, that it was folded a little more than we'd expected it to be. And then they looked at some more, and they also turned out to be folded in the same way. And we realized that this piece of RNA that was at the 5' end of the messages was actually itself split into two pieces. And so the experiment told us really two things. One was that the 5' ends of the RNA clearly were coded somewhere quite differently from the main body, but that this piece at the 5' end itself was composed of 2 parts. And so once you could do that, you could begin to map exactly where everything was being coded.
So that itself told you that there was coding in different places?
Yes, right. I mean, it just all confirmed the biochemistry, absolutely confirmed the biochemistry. But it provided the visualization that was needed. See, and the thing is, normally you do an electron microscopy experiment and everyone says, but it's just one molecule, you know. There's an awful lot of molecules in a mixture and not just the one that you happen to see on the electron micrograph. So we had both things. We had the electron microscopy and the biochemistry already done, and so we didn't really have to convince anybody of too much after that.
But just going back to the sort of the conceptualization, those must have been heady days. It was quite a long period during which you were playing with this idea that you were really going to smash the dogma.
Right.
Exciting to have that internal discussion. You must have longed to talk about it, but at the same time...
I talk to everybody about it.
I see.
I can't shut up once. I mean, I think the reason a lot of people do science is when you come across something interesting, you want to go tell everybody about it. I mean, there's nothing better than that.
What about the often talked of fear of being scooped?
Never crossed my mind.
That's nice, but do you think that was different days, or is that just the way you are? Or?
It's the way I am, I think. Right.
Because in fact, after the fact, one knows you were in competition with Phil Sharp, who was coming up with the same stuff.
Well, not really. I went to Phil Sharp's office probably 2 months before the final discovery was made. I explained my ideas on the blackboard to him, and he never said a word about it. Right. And didn't immediately then go and do the experiment that we ended up doing.
Yeah.
So. No, I think it was interesting because whenever you're really going against dogma and breaking dogma, in general, people just don't believe you. I mean, so it's not that, everyone was saying, oh, wow, great, let's go and see if we can beat him to it. I think they just didn't believe it. They didn't believe it was possible. They probably thought I was full of shit, and how it is.
So most of the
during that time, it turned out Richard Gelinas was the person who actually needed the most encouragement, because he was getting quite discouraged, because he'd do experiment after experiment and we couldn't quite nail it. And he was on a number of occasions all for packing up, and the only thing that kept him going was the fact, I'd say, well, I know we've got a really big discovery here, and this is going to be the make or break, great experiment, great discovery. And so I was, in a way, a little like a cheerleader every Saturday morning to make sure that he got up the energy to do the experiments the next day.
Well, good that you were. And it opened up a whole world. It opened up RNA editing, it opened up alternative splicing. And then 15 years later, you were awarded the Nobel Prize. Do you think it took 15 years because that world had to develop and in order for the real importance of the dogma smashing idea to come through?
I think everybody accepted immediately that it was all correct. So that was never an issue. Phil Sharp won every prize going in the intervening time because he got nominated for every prize going, and I didn't.
Yeah. Okay. So yes, it took its time, but everybody knew the importance. Yes. And then, so then your lab continued along the previous track of making restriction enzymes.
Well, actually, so we continued that on a smallish scale. We actually followed through on the splicing event. So the first thing, because my interest had mainly been in sequencing and sequences, we wanted to know what were the sequences that were required in order to be able to do this. What were the sequences at the intron-exon junction? So in between the bits that were going to be spliced out, what were the sequences? There must be some sequence there, specific sequence that the protein machinery would recognize to do the splicing. And so we set out to try to do that. And that led us ultimately to sequence the whole adenovirus genome. And so that became a project all in itself to do the adeno sequence. And then the other thing, we were trying to recapitulate the reaction in vitro by setting up an in vitro splicing system. That turned out to be a lot more difficult and complicated than we had thought. And we were in competition there. Well, we didn't really realize it at the time with Tom Maniatis' group. And we picked the Lambda promoter as a way of making transcripts in vitro, and he picked the promoter from another phage, SP6. And it turned out he'd made a better choice than we did because his promoter made really nice discrete transcripts that had a single starting point, whereas the Lambda promoters seemed to give 2 or 3 different starting points. And that proved too confusing. We couldn't then get the nice clean transcripts that we needed to set up the in vitro transcription splicing system. So he won out on that one, and there were a bunch of discoveries came from that. And we spent so much time doing the adeno sequencing that kind of distracted a little bit from the in vitro splicing work.
Yes, indeed, yeah. And then sometime later, just before the award of the Nobel Prize, you moved from Cold Spring Harbor to New England Biolabs. You moved into the industry, a small company, but industry. What prompted that move?
Well, I'd sort of done everything that was possible at Cold Spring Harbor. I was assistant director for research there, kind of overseeing the research operation. And frankly, I'd sort of done everything that I could do at Cold Spring Harbor. I mean, short of becoming director, and Jim, I think, showed no signs of stepping down, and if he had, wouldn't have picked me anyway. He and I always had a somewhat testy relationship, shall we say.
From the very beginning?
Yeah, pretty much from the very beginning, yes. Well, as soon as I said I wasn't going to sequence SV40 DNA, which he'd hired me for.
That was like day one, yes.
Yes, right. That was... He was not completely happy, shall we say, about that.
In retrospect, he must have been happy that he had you there.
I don't know. You'd have to ask him that. Anyway, so I'd been thinking about doing something other than Cold Spring Harbor. And one of the things, it occurred to me that with the advances in DNA sequencing technology, that there was a lot of DNA sequencing that it would be fun to do. And I was thinking of setting up a DNA sequencing company. And I talked to a couple of people about this, one of whom I think was seriously trying to recruit me. And then Biolabs heard that I was thinking of leaving Cold Spring Harbor and going to industry. And so, they said, well, if you're going to go to industry, why don't you come up and join us? And we talked about it a little bit, and they made me an offer that I think was just too good to turn down. And plus, I always had a great love for this company. I'd helped set them up back in 1975, and I'd been their chief consultant all the way through. And I really admired the philosophy of the owner, and I admired the way the company had done. And they were doing good research up there. So it seemed to me a good move.
So did the environment you found at New England Biolabs meet your expectations?
Oh, absolutely. I'd known the company since 1975, and so I knew exactly what I was going into. And yes, it was very nice. I mean, in retrospect, I wish I'd moved there earlier.
Do you spend most of your time there running commercial operations?
Oh, absolutely. I have nothing to do, as little as possible to do with the commerce. I'm Chief Scientific Officer, which means I oversee the research that goes on there. And so my responsibilities are all for research, for bringing new ideas into the company, for bringing in research directions that we're going to go into. And then I do my own research.
And what's the overall remit for research at the company?
So the way in which it was originally set up is anybody who came in as a researcher was expected to spend 1/3 of their time doing something that would be useful to the company. And that's pretty broadly defined, actually. And then 2/3 of the time they could do whatever they want. So that ended up mainly selecting for people who were interested in projects related to what was good for the company. So, obviously restriction enzymes, more than half our business is selling restriction enzymes. And so anything connected with that is sort of useful for the company. We sell DNA polymerases, anything connected with that is useful. And a bunch of other kinds of reagents. But the majority of the people who go there and enjoy being there enjoy the fact that they can do both pretty basic research as well as applied research and can sort of lead to the prosperity of the company.
It seems remarkably generous, allowing 2/3. Google are often proud of promoting the fact that they would give 30% of people's time to off-the-wall projects, as they call them. How do your researchers get funding?
From the company.
From the company. All of it's from the company. They're
Some people have grants. So, there's a mechanism in the US called small business grants, SBIRs. We have a few of those. We've also, in the past, we have a big program in parasitology working on filariasis. This was a deliberate program set up a long time ago when, we were reasonably profitable and wanted to put some money back into a humanitarian project. I organized a small conference. We got the people to tell us about the key diseases that the World Health Organization was interested in, and we picked one. We ended up picking the one where we thought molecular biology could be helpful and where there was very little government funding. And over the last more than 20 years now, I think 22, 23 years, we've been working in the area of filariasis. And recently had quite a big breakthrough 2 or 3 years ago, in which we and a bunch of other groups discovered that there's a little endosymbiotic bacteria called Wolbachia that lives inside the worm that causes filariasis. And if you can kill the bacteria, you kill the worm. And so this means that if you could get the right sort of antibiotic into that bacterium, then you should be able to get rid of the worm and kill the worm that causes filariasis.
And the breakthrough that you made?
We found the fact that what we were doing at the time, we had a group who were busy, this was not me personally, it was another group there, were busy looking at the messenger RNAs that were present. They were basically doing an EST project to look at the messenger RNAs that were being made inside the worm. And when they started to analyze these, discovered that some of the sequences looked as though they were bacterial and were not eukaryotic. And so when they followed that through, they were able to show that indeed there was a bacterial genome inside there, and then by sort of normal cell biological processes, you could show there was a bacterium growing inside this worm.
And how big is the outfit at New England Biolabs?
So New England Biolabs is now about 240, 250 people, something like that. We have a little over maybe 110 who are doing research.
Are they able to build up independent groups?
Yes. So what we do is when we hire a scientist, we give them a postdoc and a technician. And then if they want to get a larger group than that, occasionally if they're doing something that's especially product oriented, maybe we'll put another person into their lab to help out on that. Or they can apply for SBIR grants and enlarge the group that way. But usually we try to encourage people to set up collaborations, one group with another, and sort of expand the manpower that way.
And do people stay?
People don't like to leave New England Biolabs, right? It's very difficult to get them to go. It's a wonderful working environment, terrific working environment. Very different company. The reason is, most people set up a company because the owner wants to become wealthy. The owner wants to be the next Bill Gates. Don Combe, who is the guy who started New England Biolabs, doesn't have those same sort of ambitions. He loves research. You used to go to him and say, well, I got this wonderful idea for a product. We can make millions. And he sort of say, yeah, okay. But you go and say, hey, we got this great result in the lab today, and he'd be all over you. This was what he wanted to know about. So he set up the company with the idea of trying to make the money specifically to support research and using the products of the company to support research. He's not poor, but he's not ostentatiously wealthy in the way that CEOs of most companies or owners of most companies are.
You had tremendous success as a chemist moving into molecular biology. What do you think molecular biology needs now? What do you look for when you're recruiting to New England Biolabs these days?
I've always looked for enthusiasm and passion.
But in terms of disciplines, which?
Well, so there are a couple of areas that we're expanding into at the moment. One is the field of RNA biology. I'm a firm believer that the RNA is doing an awful lot more inside cells than we know about at the moment. One of the problems with RNA is it's not that easy to work with. You can degrade it rather easily. We know that there are huge numbers of transcripts being made within the human genome that we really don't know what they do. We are completely unaware of what they're doing. In bacteria, we know of plenty of small RNAs that are made, some of which are doing unusual things. But in bacteria in general, we don't know a lot about what RNA is doing there. You know, I'm talking about RNA outside of the normal coding RNA. So I think RNA is doing an awful lot that we don't know about. So I feel there are some really big discoveries to be made there.
But in the way that, for instance, cell biology now has some need, for instance, for systems engineers, if they get, well, they some people would say so. Other people would say that cell biology has no need for systems engineers, but it's a debating point. Is there something that molecular biology, do you think, needs from some outside discipline to help it advance, or is it?
Well, I would put it a little differently. I would say that whatever field you're in, you can always benefit from outside disciplines. People who move from one field to another always come at the new field with a different underlying philosophy of how to do science. And that's a good thing. People, when you move from one field to another, it's very easy to ask stupid questions and not be made to feel stupid, because people will say, oh, well, you know, they weren't trained in the discipline, so they don't know any better than to ask that question. And it's often the stupid questions that really allow you to make progress. Because then when people are trying to answer them, they suddenly realize they don't really know the answer to some of these stupid questions. And so then you can go ahead and provide the answers. And sometimes they're not what you expect.
Well, perhaps that's a nice end point, the thought of recruiting a few stupid people to New England Biolabs.
No. You want smart people from different fields. So this is a good thing to do. Yeah, no, stupid people in general don't help you all that much.
Okay, well, thank you very much indeed for taking the time to speak to us.
Okay. Good, thank you.